Dober večer in lep pozdrav vsem ljubiteljem znanosti in prijateljem znanstvenih večerov, zdaj že tradicionalnih na Zemonu. Danes imamo veliko čast, da je med nami dr. Tanja Dominko, Slovenka, ki je svojo znanstveno pot nadaljevala in jo še nadaljuje v Združenih državah Amerike. Tanja je diplomirala in magistrirala na Univerzi v Ljubljani in se po magisteriju preselila v Združene države Amerike. Slovenija je očitno postala pretesna za njen način razmišljanja, kot boste kasneje iz predavanja videli, in je potem doktorirala na Univerzi Wisconsin v Madisonu. Potem je pa svojo pot nadaljevala če se prav spomnim, Oregonu, kjer se je ukvarjala z enim izjemno zanimivim področjem, to je kloniranjem primatov. Potem jo je pot zanesla na Worcester Polytechnic Institute, kjer ima danes raziskovalno pozicijo in kjer je potem tudi ustanovila svojo lastno firmo in kot boste videli, se da moderno znanost tudi dobro tržiti. Danes mislim, da boste imeli enkratno priliko, da slišite najmodernejše področje znanosti o življenju, life sciences. To je problem kloniranja in seveda uporabe teh znanstvenih dosežkov in rezultatov v korist človeštva v namene regenerativne medicine. Da ne bom povedal kaj narobe, ker nisem strokovnjak z tega področja, bo najboljše, da predam besedo kar naši današnji gosti in predavateljici. Tanja, prosim, izvoli.
Hvala lepa. Hvala lepa. Hvala lepa. Hvala Univerzi v Novi Gorici in profesorju Zavrtaniku za vabilo. V 18 letih, odkar sem v Združenih državah, je to moje tretje predavanje v Sloveniji. Bi jih rada še več imela in nadaljevala, upam, pot, ki sva jo s profesorjem Zavrtanikom poskušala zastaviti med mojim laboratorijem, ne samo mojim, med znanstveniki na drugi strani morja in Univerzo v Novi Gorici, in sem z velikim veseljem prišla na obisk in upam, da vam lahko pokažem ene par novic na področju regenerativne medicine, ki v svetu postaja vse bolj pomembna. Jaz se opravičujem slovenskemu občinstvu, ker bo predavanje v angleščini. Moj strokovni jezik je tako majhen na tem področju, da ne bi rada razmesarila svojega predavanja v slovenščini. Bom z veseljem odgovorila na vsa vprašanja, če bom znala v slovenščini, lahko debatiramo po koncu predavanja ali v slovenščini ali v angleščini. Thank you again very much. Cloning and embryonic stem cells in regenerative medicine is a field that in the past years has gained a lot of experience, but I think it's really important for some of you to recognize how does one become involved in the field of regenerative medicine. There's no school that says, I want to be a specialist in regenerative medicine. I just happened to start right here, about 100 miles away, at the University of Ljubljana, and graduated at the veterinary faculty, which per se was not dedicated to developing human therapy, but nevertheless connected with biology quite a bit. And after I got my master's in reproduction and obstetrics, I became involved, again, another step in my path, how I became who I am today. It's really early embryonic research, and this was in early 1990, when the field of early embryology, or embryology in general, was really not developed in Slovenia at all, not even in former Yugoslavia. There was really no place to go. So after starting as a clinician working with embryo transfer in animals, in horses and in cows, I really ran into a problem that I didn't know enough about embryos. I didn't know how to make embryos. I didn't know enough of embryo biology to be as successful as I wanted to be. So I decided to pursue my studies by applying to a graduate program in University of Wisconsin-Madison. I was accepted. I was very, very lucky at the time. And that's really where my path to understanding embryology began. I did my PhD thesis really focused on understanding, and I do have help, on understanding what is required to get from an oocyte, this is a female gamete, to fertilization and support early embryonic development to develop a stage of an embryo that eventually will be transferred into an animal. And right here you have to keep in mind that this was all done with applications for agriculture primarily. And cloning that is today successful in cattle, pigs, mice, rabbits, goats, horses, dogs, cats, and probably some species I didn't even mention, really originated from a desire to propagate a specific genotype. For example, if you have a really good racing horse, you wanted to make another identical animal, hopefully with the same potential. So cloning started because of agriculture. Cloning didn't start with an idea that we're going to eventually use it in humans. So this is where the beginning is. And today, a lot of scientists that are involved in the field of regenerative medicine came from very much the same environment that I started in, veterinary medicine and animal science. As you heard just before, I moved on after I got my PhD to Oregon Health Sciences University, where they were working with rhesus macaques with the primary intent to develop animal models for studying human disease. For example, we had an animal that had a human condition that some of you might recognize. It's called Retinitis Pigmentosa. It's affecting eye. It's an eye disease. And there was only one animal that had that condition. And in order to study any therapy for any condition, you have to have a population of animals. And the only way that we could make more of the very, very same genetic makeup was to try to clone these animals. So in addition to agriculture, now cloning became one of the tools where you could really try to produce models in biomedical research for testing human disease. You saw on the previous slide that this cloning technology, even though it sounds very, very science fiction-like, it was successful in very many species. However, after working on this project for almost 4 years, we were repeatedly unsuccessful. And this is not a lecture that anybody wants to give about how you worked for 4 years and you really didn't accomplish much. But nevertheless, that's what happened. So repeatedly, after we were creating these embryos by nuclear transplantation, we noticed that they were behaving very much like cancer cells. They were very abnormal. If you look closely, and I don't know if we can dim the lights because you could see even better, this is a one-cell embryo, and it's trying to divide through its first division. And if you look closely enough, you can see that part of its nuclear material, genetic material, DNA, is being pulled to one side, and on the other side, instead of an equal picture, it's being pulled to two sides. So instead of ending up with 2 normal cells, you would end up with 3. And that is not normal. That's how cancer cells sometimes divide. Not only did this happen, but as you go further through 2-cell stage, 4, 6, 8, 8-cell stage, these abnormalities became even more pronounced. So we would end up with an embryo that you saw before. Let me see if I can do this just for illustration. That would essentially look like this. If you looked under the microscope, the embryo would look like that, and you would say, this is a really nice looking embryo. But it turned out, if you looked inside and labeled DNA, DNA is in blue, you could see that only 2 cells in this example contained any genetic material. So clearly, this was a process that in primates was not going to be as efficient as it was in cattle, pigs, and in other mammals, and that was really very surprising to us. We'll get to this, and as of today, cloning a live animal in primates, non-human primates, has not been successful. And I imagine you have heard rumors 2, 3, 5 years ago that people have been already cloned. It's simply not true. It just hasn't happened yet. But during this time, when we were really trying to discover how to make this happen in order to achieve biomedical models, two publications came out really in 96 and 97 that showed first important scientific possibility. And that was when Campbell and Wilmot in the United Kingdom, actually in Scotland, managed to clone the first sheep. So what does clone mean? Clone means that they took an egg from a black-head sheep. They removed its nuclear material, so what you have now is only an empty shell. It doesn't have any genetic information, and if left alone, would result in absolutely nothing. And then they took a cell from mammary gland, any regular house variety cell, and I'm going to call them somatic cells because they're adult body cells, and they united these two now. So now genetic contribution was not coming from the egg anymore, but genetic contribution was coming from this animal. And after they united them, produced an embryo, transplanted this embryo into a sheep, they got Dolly that was genetically identical as her mom, which really wasn't mom anymore, right? It was a twin sister, except they were different age because it was an identical twin. But nevertheless, for the first time, it really shifted the biology as we knew it, saying that there is no such a thing as a final differentiation. I said the cell goes down the certain path and it can never turn back in time and recapitulate development. Well, this said that that's not true. It doesn't normally happen. But if you take the cell and you put it in the right environment, in this case, environment being this empty eggshell, it can go all the way back in time because it had the ability to produce the whole animal. That was a critical discovery that is eventually going to lead to applications in regenerative medicine. But a fact that there is no terminal differentiation anymore as a dogma was broken once and for all. And that was about 10 years ago. To just give you an idea how procedure is done and how it works, it's really a biopsy that is taken. Doesn't have to be any bigger than the size of essentially a pin when you compare it to a coin. This is a piece of tissue that can be obtained from skin, from any easy biopsy kind of site. This piece is put in culture, and you can see if you grow them, these primary explants in culture, you can establish a really nice layer of usually fibroblasts that can be kept in culture. Before nuclear transplantation or cloning experiment, you have to detach these cells because they stick to the surface. You put them in suspension, and they're essentially ready to go. Well, the next step that I mentioned before, and I didn't spend much time on, is really in order to take advantage of this new genome from the cell that you really like very much, from an animal that you like very much, you have to remove the genome from the mammalian egg. And here is the same image after staining with a DNA-absorbing dye that can be visualized in the UV light. And we do this in order to make sure, to verify that we have removed DNA from this egg and that only shell remains. So what we do, these are micromanipulation tools. It's all done under a microscope. And the size of this egg is about 150 microns, to give you an appreciation of the size. So this has to be done under the microscope. DNA has to be removed, verified that it's in here, so you don't have any interference from the other side. The second thing, now we have to introduce our cell of interest. The cell is now in here. We have picked it up from our cell suspension. You go usually through the same hole that you started when you removed egg DNA. You insert the cell right underneath the zona. The structure over here is called the zona. So the cell adheres nicely to whatever is left of the shell. And here's another cell waiting for another oocyte to be processed, essentially the same way. That's not in itself enough. If you put these units together, you haven't accomplished anything biologically significant yet. So we line these units now that have a little somatic cell next to the eggshell into electric field and apply pulse across the electric field, which induces membranes to fuse together. So now what we accomplished, We introduce now this DNA, genetic material from our cell, into this empty shell, and this structure has a chance now to develop. To give you a sense how this looks in real time, I think it's kind of cool to just watch it in real time. Maybe it's not. We might have to do this at the end because I'm not sure how this is set up now. To play the video, this is really a video that shows you everything in real time, but we can move onto the next one. And if you're lucky, you can produce blastocysts. This is about day 6, day 7 structures in embryonic development that are produced after such a procedure. So essentially you see that you can artificially make twins because half of the embryo extrudes itself through the hole that was made during the micromanipulation procedure. Well, all that worked fine for cloning purposes of agricultural animals, and I told you, well, it really does not work in primates. So why do we still keep on talking about use of embryos and potential that embryos have in order to advance regenerative medicine? And to make my talk a little clearer, our goal is really to figure out how to regenerate amputated limbs. It's a lofty goal, we all understand that, but the goal is really to remove scar-forming tissue and allow the leg to hopefully extend itself one more time. The second very important thing that happened after Dolly was cloned and the cloning determined that there is no such a thing as a final differentiated cell, the second paper was published a year later that said not only is that true, but if we take now this embryo that we have made, either by nuclear transplantation or in vitro fertilization, there is a structure inside of this embryo, and I can show you. This would be this structure in here that contains between 10 and 20 cells that really have this fabulous potential. So what they did, they took human in vitro fertilized embryos, they dissected inner cell mass. The structure is called inner cell mass, and it's the one that usually forms the fetus. All the cells that surround it are tissues that will form placenta in the end. So this is really the cells that will contribute to all the tissues of any animal organism. But even more importantly, if they isolated these cells, they put them into a petri dish, they could grow them for a very, very long time. Meaning that these cells, not only because you start with 20, all of a sudden they had this potential to self-renew and to grow extensively. And even more fascinating, where regenerative medicine really started, is the potential of these cells. If you expose them to the right conditions in culture, they have the ability to now become neurons. A little later, they discovered, now if you change the conditions that you put them in, you can develop cardiac muscle cells. Similarly, you can develop all the lineages of the blood lineage. So all of a sudden, this little part of an embryo, regardless of how you create it, with nuclear transplantation or not, became very, very important because of this potential. Saying essentially that if you had an embryo and you needed tissue for transplantation into a patient, functional tissue, you could develop this tissue from embryonic stem cells. And that's where these two fields essentially came together. And the third equally important discovery, which is probably the oldest but was never appreciated enough, is that stem cells exist in our bodies normally. It's not only embryos that contain stem cells that have this wonderful potential. Stem cells exist in brain, they exist in the heart, in bone marrow. Bone marrow transplantation, we all know, has been around for decades. It's not a new thing. But why does it work? It works because there are pockets of stem cells in the long bone that are true stem cells, that have the ability to produce not only red blood cells, but white blood cells and platelets and everything else. And they're called adult stem cells. The problem is that they're not quite as magical as embryonic, because the bone marrow cell, even though it's a stem cell, cannot go back in time and remember how to form neurons. So they're much more lineage restricted. Blood stem cells form different types of cells in blood. Heart cells really pretty much develop new heart tissue, but they also don't know how to make blood. They don't know how to make neurons. And the same, brain stem cells, just about impossible to make blood from. So there's the critical difference. The embryonic cell can do anything and everything. These cells can do a lot, but they are a lot more restricted in their potential. And of course, even though this is being used extensively today, especially bone marrow transplant, more recently heart cell development as well, embryonic stem cells still became the target of choice to pursue. Because of its ability to do anything and everything we want, all of a sudden, a list that is still expanding of conditions that are caused by cell death became prime targets for development of cell transplantation. And they pretty much belong to two categories. One is degenerative disorders, and we're all familiar with those. Parkinson's, Alzheimer's, Huntington's, diabetes. All these diseases have one thing in common. The cells that are producing very important biological molecules for regulations and functions are dead and are not coming back. The tissue doesn't know how to regenerate if your islets in your pancreas die. That's why people have to take insulin for the rest of their life. So degenerative disorders like this are one candidate. And the second one is really any massive tissue loss, like amputations, like massive tissue trauma after car accidents, after any kind of accident that you can imagine where a majority of, most of the tissue of a certain limb is lost. We know that that tissue doesn't regenerate anymore. What happens in mammals, unfortunately, is the scar covers the wound, and that's pretty much the end of the story. So now all of a sudden it became possible to ask a question. If we can have stem cells develop bone and muscle, can we use their ability to now reconstruct these tissues in living human beings? And that's where all these areas really came together, knowing that stem cells exist, number one, knowing that we can make them from skin cells, which became very exciting because for the first time it offered an opportunity that you can have your cells for transplantation built from you and you can have your own. No patient matching anymore. It for the first time became even a thought that maybe customized medicine that is patient-specific would one day be possible. And that's why embryonic stem cells gained so much popularity. Until this happened. I worked for Advanced Cell Technology briefly for a year in year 2000 when they decided that they were going to propose they called it therapeutic cloning. The principle is exactly the way I explained it before. What you do, you take a patient with a degenerative disorder or injury, you take a skin cell, you produce an embryo using nuclear transplantation procedure, you isolate the inner cell mass, you put them in culture, you develop whatever the problem is in the patient, and you can give it back to the same patient. It's immunologically compatible transplant that has clearly many, many advantages over what is currently available. But that was one huge problem that came with this approach. And it was, number one, where is this egg gonna come from? Where do I go to get a human egg? They come from women, and that's not an easily accessible material. We all know that. And the second, which is even more controversial, and it remains controversial today, I think for good reasons, is that if you were to engage in this procedure, you would be creating an embryo in order to literally kill it at the end. And that's where government had quite a bit of a problem discussing embryonic stem cell research in those terms. So creation of an embryo just for the purpose of extracting embryonic stem cells was not good enough to convince the country, I'm talking about the United States now, to allow this procedure to be used for medical purposes. And if you can't do that, there is no way, as we knew it back in those days, that you would be able to produce immunologically competent cell transplant. Because if you cannot make an embryo from my cell, I can never have my own therapy. The promise, however, was huge. There's another big advantage that embryonic stem cell transfer allows. And that is that the cell and its ability to grow in culture in dishes offers a great opportunity, not only to investigate what it can become and give it to the patient, but because its genetic material becomes accessible. What does that mean? Well, we can take advantage of that in diseases that are really regulated by genetic mutations and genetic problems. If we have a patient that has a certain disease because a gene is wrongly regulated, what we can do, we can correct this problem in the cell by manipulating its genome, and then return healthy cells to the patient and hopefully eliminate the condition. So it's not only that they can form all tissues, we can actually fix problems in DNA if we needed to in order to create therapy. Stem cell is really not all that special. I would want to argue that is the least special cell of all. What really defines it is that it has indefinite self-renewal, so it always knows how to divide in such a way that it retains its pluripotency, potency to form all the tissues. At the same time, some of the cells that are produced by cell division are a little further downstream and are able to follow this differentiation pathway. But nevertheless, this cell is like the ultimate mother cell that retains all the information. And why is it the simplest cell? In my mind, is because it can really become anything. So it's a very, very basic cell type. It doesn't have an imprint yet. Well, getting from here to here, politics is only one thing. Only one of the things that can convolute everything. And the second one is, and I'm glad I'm not giving this talk in the United States, but it's people who are in charge that really very judgmentally decide what is morally right and what is morally wrong. And the United States, unfortunately, has been left behind in embryonic stem cell research quite a bit because of government really not listening to compelling arguments. Singapore, Australia, United Kingdom, there's a number of countries that have taken a different approach and allowed this field to really develop for millions of people that suffer from degenerative disorders. Okay, so if we can't use embryonic stem cells, how are we going to go about making this ever happen? Well, can we do this on demand without embryos? I believe we can. I'm going to try to convince you in the rest of my talk why do I think we can actually do that. You all know salamander. Močaradi, nevím, kdo by še bil, kuščari, yeah. Salamanders have this great ability that if you cut a leg off, over a period of about 30 days, this animal can regrow slowly the full functional limb back in its entirety. The limb is the same size, it functions properly, it has all the attributes of a mature limb. And it's a great model where regeneration is studied. In humans, on the other hand, any trauma of this magnitude will always result in a non-functional scar. And I would expect that that would make everybody angry. It makes me angry. Amphibians can do it, but human can? There has to be something that we can learn and take advantage of this little critter and help us figure out how do we do this in humans. They can do it. We can do it. We can't do it. But what's even better, we know that the cells that make this happen are connective tissue fibroblasts. Though fibroblast is probably the most ubiquitous cell in any mammalian body. It's in every connective tissue. And in salamanders, the fibroblasts are the cells that go back in time. After injury occurs, They go back in time and they become essentially a stem cell right there at the injury site. And not only do they become a stem cell, but by becoming a stem cell, the tissue where it happens is telling them, well, a leg is missing, grow a new one. Don't grow a nose, don't grow, you know, a liver. No, this tissue right here has all the information to tell these little tiny bump on top how to produce this again. And we have plenty of fibroblasts. So the common link is here. So how do we take advantage now? But in our case, fibroblasts don't know this anymore. They have the memory. They just don't remember. The information is all there because all cells are the same. Genetic information is the same in every cell in your body. And yet, when this happens in human, fibroblasts don't know how to do this. They just cover the wound and form a fibrous scar. So we had a common target and we said, well, let's see if we can make this happen. So how do we go about it? Well, we know that in nuclear transplantation, you remember I was talking about the shell that is left behind that the empty egg provides. Well, that yellow component inside of the egg obviously then contains every factor that is required to make stem cells, right? Everything in here. If you put a cell in, it has the ability to grow an embryo, make the stem cells, and make the whole organism. So if we could figure out what components are there in the egg, what is it that makes this happen? If we can dissect the system in such a way to gain information, maybe then we can do it in a dish, in a laboratory, and we don't have to use this embryonic intermediary. Instead of, by learning what this is, maybe we can shortcut this ethical, unaccepted circle and make stem cells in a different way. Well, where do you get eggs to do that from? If you remember, I said egg is about 150 microns. That's very small. There is no source in the world that would provide you with enough material to start looking at these components. It's so small. You need so many. It's physically impossible. And again, amphibians to the rescue. Amphibians such as frogs have been used for research for decades for different purposes. But a very nice thing about frogs is they lay a lot of eggs. The eggs are huge in comparison to mammalian eggs. They are over a millimeter and a half in diameter. And every animal can produce enough eggs that if you induce 5 animals to lay eggs, you can produce about 20 milliliters of that soup that we know contains everything. Because the factors between the frog and the human are the same. That has been determined. Because the frog egg cytoplasm, which has been centrifuged in here, has the ability to do the same to a human cell as a human cytoplasm has. We know that for a fact. So we chose the frog. We take eggs, we centrifuge them down, we extract everything that is extractable in the cytoplasm, and we introduce this extract. We still don't know what it is. We just know it's good. And we put it in our fibroblast, and in a couple of days, we start getting morphology, appearance of our fibroblast. You can't see it here, but if you remember the fibroblast culture, they don't look like fibroblasts anymore. They look more like beginnings of stem cell colonies. We put them back, stimulate them to re-enter the cell cycle, and they remain the same morphology. Well, okay, they look like they changed. Yeah, big deal. What does that mean? Well, we know that there are 2 things that need to happen in order for cell to lose its memory. For a skin cell to become a stem cell, at least 2 major events have to take place. One is chromatin remodeling. We developed, and I'm not going to bore you with details about this, let me just explain that we have created a transgenic cell line, a fibroblast cell line. We introduced a protein that glows green when exposed to fluorescence. So, and it's always located in the nucleus of a cell. So the premise would be, if anything is happening to the nucleus, we should be able to see it. Chromatin remodeling is required to really strip genetic repression off of a skin cell. If I make it as simple as I can, if this is fibroblast DNA, this is the part of it that is making it a skin cell. Everything else is messed up, it's covered, it's repressed. Nothing is working, just this part is working. In liver cell, maybe it's this part working. Well, in a stem cell, everything is removed. All the repression is gone, so everything becomes open and active and acceptable. And we monitor this by this green protein that fluoresces in the nucleus. And after we introduce our magic extract, we can see that within 105 minutes, most of the nuclear protein, over 90% of the nuclear protein, disappears. And this happens to be histone H1, which for those of you who are familiar with biology, you know it's a tightly regulated histone that packs nucleosomes into a structure that DNA has. So this was a very good sign. It was telling us, we know that whatever it is in there, it has the ability to clean everything that's on top of DNA and open it up. That's a good sign. So, but again, like anything else, it's a step closer, but it's not where we need to be. And the second thing is, not only do you remove the information of DNA, but this cell has to forget what it was. So it can't be a fibroblast anymore. So if you look for a protein in green, FSP, that is expressed in fibroblast, after electroporation, it starts disappearing, which is another good sign, saying, you know, this fibroblast doesn't know that it's supposed to be making a lot of this green stuff anymore. So it's losing this memory, which is good. Even better, at the same time, it's starting to remember what's really important and has been described as critical in embryonic stem cells. And those are SOX2, OCT4, MSX, and nanog gene expression that are all localized to the nuclei because they're all transcription factors. When embryonic stem cells were first discovered, these were the 4 markers that described them as stem cells. If you have this green here that co-localizes with your DNA, it's localized nicely in the nucleus, they said you have stem cells because there's no other cell in the body that gives you a picture like this if you just take it out and put it in culture and label it with immunocytochemistry. So this was really 2 pieces of good information. Number one, what it used to be, it's not anymore, and what it wasn't, it is becoming now. We have no expression of these embryonic genes in our control. Fibroblasts that didn't see the magic extract from eggs ever, but we get all the expression. Again, this was too exciting. This was too exciting. This was never described before. So we said, there has to be a problem. And we started chasing problems without even realizing, well, maybe it's not a problem. Maybe it is possible. But what happened, because at one point we weren't able to repeat these experiments, we started doing time experiments. In biology, way too often, we expect things to happen quick. I put the cell in a dish and I give something to it, and voila, in 6 hours, something needs to happen. Well, in this case, that's not true. It turns out, and this is just an example of one specific gene, that this gene shifts in its expression of molecular weight 7 days after you started this treatment. And today we know that there are certain genes, like OCT4, it takes about 2 weeks to start expressing. So this taking skin back in time, if you remember that frog cut off leg, back in time takes time also. It's not an instantaneous event, but it is very predictable and very repeatable. Okay, so now again, we're back to the beginning. We know extract can do these things. What is it in the extract? We started with simple fractionations based on molecular weight and based on solubility and based on a number of things. And we isolated 2 very specific fractions that contain the activity they still are not defined. We still are not quite sure what is doing it, but we know that this one, and this one, and this one, and the first one can't do what I showed you a stem cell should be able to look like. So now we have two good targets that we are chasing their components through a lot of protein biochemistry and RNA analysis. So the way we test now, I told you they look like stem cells. It appears they are expressing the right genes, which makes us think they're stem cells. But that, again, is not enough. What happens if you want now the cell that you say is stem cell to become something else? Remember the slide? A true stem cell, if you put it into neuron conditions, will follow neuron development. If you put it into muscle-stimulating conditions, we go into muscle. So we ask a next logical question. Do our cells have any of that ability? And until now, we had demonstrated at least 3 different tissue capabilities in our cells. If we expose them to dexamethasone, BMP4, and a couple of other factors, they have the potential to form bone. Again, we started from a skin fibroblast. We can't forget that. This is a cell that was never supposed to be bone. They can make mature neurons. These red long lines are labeled with a protein that is only found in adult neurons. Again, it came from the skin. It's not supposed to have it. And the third one is fat tissue. Skin fibroblasts don't accumulate fat. But if you put them into conditions, they can form adipose tissue. So we now have indication that at least under right conditions, our cells seem to have some ability to be pushed where we want them to go. So they're clearly not just simple skin cells anymore. The ultimate test, and you must have seen this in embryonic stem cell biology, is really everything I told you so far. It's all in a dish. It's all in the laboratory. It's not in the patient. It's not in a living system. So even though this is very promising, it is still not answering the question, will this function in a living tissue? So a common test is you inject your cells into a SCID mouse. It's an immunocompromised mouse model that does not reject transplants. And after you inject your cells into this tissue, if it forms tumors, it's a good sign that in vivo now, in a living organism, these cells have this potential to become a lot of things. In this case, it's really a cartilage tumor. And in this case, we developed a tumor after our, again, skin cells that looks like a primitive fetal liver. And these tumors were developed in a leg muscle. This was not taken from anywhere else. You see it's nice and contained, but you can clearly see from histological sections that it looks like a fetal or embryonic liver. So clearly, our cells have been manipulated in such a way, and we know where the magic is contained now. And I wish I could give you the final answer, but I can't, because then I can't come back and give another lecture. We know a piece. But at least we have identified components now that we feel very comfortable with. After they are defined, we might be able to now chemically convert everybody's cells into these magic cells that could be used for cell therapy. And nothing happens in a vacuum. And this is the proof. Yes, I always give the talks, but if it wasn't for some of the people in my lab in Worcester, if it wasn't for people at University of Massachusetts Medical School, Tulane University, University of Louisville, California, and my dear friends that give me money so I can do my research, none of this would happen. And I want to leave you with this slide as equally important to everything you have heard today. There is no problem today in life sciences, and probably across the board, that can be solved by a single person. And if this is possible, it should be taken advantage of. It has to be taken advantage of so we can utilize the expertise of a lot of different people at the same time on the same project and come out with even as crazy a proposal as you have heard today, essentially making a skin cell a stem cell for you. Thank you.
Tanja, maybe we take a seat for...
Mi boste pa kaj vina dali, pa kar vodo.
You'll get one a little bit later. Okay, I don't know, maybe we should continue in English, because I see that there are people from various countries in the audience, and I hope that the Slovenian part of the audience does not care if we continue maybe in English, but we will take questions, of course, sure, in both languages. So thanks again for a very nice lecture, and it was really for us non-specialists on the limit of the
biology.
Well, I mean more science fiction, and yeah, but it's amazing. I mean how big potential is in this type of research. But maybe we can go a little bit back in time, and I'd like to ask you, what was your main motivation, or if you want, the trigger that you left Slovenia? Because nowadays we have a lot of very bright and promising young people, and unfortunately they believe that they can see all the science in their small country or even small town. So what was the motivation, or how did you discover that you cannot continue in Slovenia if you want to do this type of research?
At the beginning, it wasn't ambition that I needed to go any place to learn anything. I was forced into making some kind of decision because I did not feel competent doing my job anymore the best I could without having a more detailed knowledge. We started doing embryo transfer at university in Ljubljana back in late 80s, and we had really poor success. The rest of the world was doing it left and right and publishing papers, and we could barely get a pregnancy. I said, it's not because we're stupid, but there's one thing is reading the publication and then actually doing it. It's not the same thing. You cannot learn science from reading only papers. So I decided I absolutely had to go and learn more, even though at the beginning the idea wasn't, I'm going to go, and here we are 18 years later, and I'm still there. I didn't come back. The idea really was to advance my knowledge, to find the best place in the world that I could do it at. Even if they said no, that's okay, I'll find the second best place. But I was really driven by the desire to know as much as I could know.
Okay. Maybe another question. How did you end in Oregon? I mean, doing the cloning of primates? Was this by chance, or?
I think everything in life, most of things in life happen by chance, meaning you have to be in the right place at the right time. And that was part of my story as well. I worked in a strong embryology lab, and the lab across campus, on the other side of campus, was offered to move to the primate center. 20 people were moving. The whole laboratory was hired by the primate center, and they really wanted somebody who had more of the animal side experience, and we were all friends. So they said, do you want to come with us? And I said, yeah. And we packed and we went. It's really through the people that you work with, it's the connections you make, and the interests that you're willing to sometimes risk to explore. Even though I never worked with primates. I'm a veterinarian, but I never worked with primates. I worked with cows. So this was like a whole new intimidating factor. But you know what? Biology is biology. And today I'm in regenerative medicine, and I'm not a doctor. you have to be willing to take a risk sometimes. Go with your gut instinct. Yeah.
But did you expect the problems with the cloning of primates? I mean, because for a, let's say, non-biologist, a cow or a primate, it's not a big difference. Huh? So
we really didn't. We did not expect it at all. And after a few failures, quite a few failures, of producing anything that looked remotely transplantable in terms of an embryo, we had to step back and say, we can't follow the philosophy that if we do 1 million of these experiments, one will work. That's not how science should be done. So we said, that's a waste of time. Let's go back. Let's start examining every step at the beginning. Let's first fuse the cells and look at them. Let's let them divide once and look at them. Let's see if we can find where the problem is. And that's how we stumbled onto this incompatibility between the machinery that is required for DNA separation. Just was not talking to each other. The egg part and the cell part was not communicating properly and was making this gallery of horrors, we called it, because not one embryo looked normal. That is not to say that cloning primates is impossible. Just like regrowing arms is impossible. It's not. It just hasn't been done yet. Somebody will eventually figure out how to do it. Nothing is impossible. We just didn't do it. We learned a little bit why not, but nothing is impossible.
So you also founded a company in the United States, which is, at least in Europe and in particular in Slovenia, it's not very often that a scientist founds a company and then which survives and it's successful. I mean, how much do you have to invest? I mean, not just in money, but also in intellectually and to create such an entity.
I think it was my ultimate adventure in life. It was. I took it very seriously later. I started it in my pajamas at 6 in the morning on the computer. I was so angry that I could not do the research that I presented to you today because nobody would allow me to do it. There was always something more important. And I saw the potential. And I wanted to explore it. And I said, the only way I'm going to be able to do this is to have my own company. And literally, in 10 minutes, I had my own company. I had a name. They had my credit card number. But I had a name, and I became an entity that now could compete with universities for funding. I was an entity that could employ people. I finally had an organization that would allow me to follow what I wanted to follow. So it's an adventure that doesn't come cheap emotionally, and it's not guaranteed success. And a lot of people here will definitely agree with me because you don't know. We're still not making any money on what I showed you. We still don't have a product, and we've been around for a few years. But I still believe that at the end of the ends, this will be used for medical intervention in the future. So I'm still willing to pursue my dreams. It was difficult in the beginning financially because I spent my children's college funds. I have spent every saving that I ever had in my life. I sold the car. I still have one, but I sold the other one. So there's not a penny to my name. And yet, I think it was worth it. Because now finally it's paying off. We have done enough research to demonstrate that they need to take this approach seriously. And that's why the government is funding a lot of our equipment, a lot of our research, and promise that one day this will work.
Maybe another comment or question. Well, these ethical problems, when you talk about cloning, all the time you are bumping into the ethical problems, and some countries do allow research on stem cells and others do not. But do you think that this fact that in the United States it was not allowed to do it actually triggered you to search for new approaches and Because otherwise, if something is easy, you will probably follow that path. Huh?
True, very true. That was one of the factors that it became almost impossible. Research on embryonic stem cells in the United States is allowed on existing stem cell lines. They exist, they were created from publications that I showed you. There is 16 embryonic stem cell lines that were created before the government said no more. Those are okay. So we can do it, but you can't make new ones now. I can't make my own, I can't make yours, I can't make anybody's. But definitely when you're in the situation of, when they push you into a corner, it's very easy to get creative. You just have to. You have to, otherwise you're gonna stay in the corner and not move anywhere anytime soon. So I agree, I never thought about it, but I think it's a good point. If they just allowed it, maybe we wouldn't be where we are today.
You'll miss something. Huh?
Yeah. We'll be doing something else. Yeah, I don't know.
Okay, maybe I should ask the audience for comments or questions, and please speak in any language we understand.
Hello.
Hi.
So when you introduce the skin cells to this frog cocktail, so you have to wait for some time so they stop remembering what they were and so on, unwrapping the DNA and so on. Right. Does this process goes faster with the semi-stem cells you mentioned at the beginning that humans also have, like in bones or in brain? Does this process goes faster with them?
See, nobody has done that experiment, because once you take a stem cell out of an environment where it exists, it already has a lot of these characteristics that we are trying to artificially induce. But there are recent publications that I didn't talk about, and I have to recognize, that achieve identical results that I have shown you by introducing into skin cells viruses that carry these 4 genes. So if you artificially put these 4 genes into any cell, it will essentially become a stem cell, but it's virus transmission. It's a whole different story. They showed very similar time pattern. But if you think about embryonic stem cell, in nature it doesn't even exist, so we don't know how long these cells really take to become what they are.
Maybe another question is, you are not also allowed to import stem cells into US?
You can do a lot of things with private money in the United States. So if it's not government-funded research, if you have a corporation, General Electric, it doesn't matter who they are, and they give you $5 million, you can do any research you want. It's not illegal to make them. It's federal funding cannot be used for derivation of these cells. But it comes with a catch. And the catch is that because it is not illegal yet, that doesn't mean it will not be illegal tomorrow. So if they give you the money and all of a sudden they criminalize the procedure, not only as an investor have you lost your money, but your people might be going to jail. So investment into embryonic stem cells has gone way down because people are nervous that the government one day is going to say, not only you can't use federal money, but we're going to make it illegal to do this research, which would be a serious, serious issue. But nobody knows.
Thank you, Tanja, for this very nice lecture. I have one question. You mentioned mainly that the purpose, or let's say, how can I say, the sense of your work is because the soldiers are coming from Iraq without legs or hands. So that means that actually their tissue was healthy. What do you think in case of when amputations occurs because of the illnesses, like diabetes, it is possible to regrow then the leg, or let's say?
I think it's possible, but I think you have to deal with the problem of diabetes at the same time. Because if you don't remove the cause, in case of diabetes, you can induce the leg to, if we learn how to induce the leg to regrow, the same thing may eventually happen again. So I think those problems will need to be fixed on more than just one level for diabetes, especially. But for example, right. The only link that we have so far that we can say could be treated at the same time is genetic problems. Because we could go into these cells and either cut genes out that are bad genes or suppress them and prevent the same thing of happening again. But that's a very good point. If you can't eliminate the cause, if it's such as in diabetes, it doesn't in the long term do much good effect.
So one approach that seems to be interesting, at least it's advertised also in Slovenia now, it's isolating stem cells from umbilical blood, such as the embryonic stem cells. So do you think that this approach would actually help remove all the problems for people who are born from this point on, if you just freeze their embryonic stem cells and just grow in every organ that you wish from those?
Right. Well, that question could be answered in two ways. Embryonic stem cells, essentially, and I wish I had a couple of more slides for that. What we can do today with an embryo, assuming you go through in vitro fertilization or something assisted, where the embryo is actually in a dish, what we can do, we can cut the embryo in half or remove a few embryonic cells and culture them in a way that will form embryo again, like forming identical twins. And then I know this is not politically correct, what I'm describing, but then what you could have is one embryo gets implanted and results in a baby, and the other embryo is frozen and available possibly in the future for derivation of the same genotype embryonic stem cells. Are they doing it? Yes. There are clinics that offer that service because of the promise of embryonic stem cells. The second one is for babies born, and that's what triggered it, is really umbilical cord blood. Is the program Saving Umbilical Cord Blood from Newborn Babies is one of the biggest programs in the United States because, again, is one of the tissues that contains very potent stem cells. They're not, again, embryonic kind of quality, but nevertheless, they are stem cells, and they could be used for babies when they grow up. God forbid they had a problem. So those are two things. But for the rest of us, we're toast. There's not much that we can devise to save ourselves. That's why we're working hard for the rest of us.
Actually, I have another one, if I may. It's a bit more a technical one. So you mentioned the chromatin remodeling that has to occur for the dedifferentiation. There is a recent paper that H2AZ histone is actually involved in the cancer development. So that it seems to be really like it's hard to distinguish between these two processes of having dedifferentiation of a stem cell into something or the cancer progression.
That's an excellent question. And again, next year I will tell you, hopefully, the answer to that story in another lecture, which I expect an invitation for.
Surely.
No, but in all seriousness, the components that I show you very briefly, those two fractions, if you remember, we have tested individual components that we have identified, and we can replicate pretty much a lot of the characteristics of stem cells, but they don't make tumors. So I think by chemically defining this change in memory loss without reactivating the telomere, telomere is the segment of the chromatin, for you who don't know, that really shortens. Every time a cell divides, it becomes shorter and shorter and shorter. That's why we age, because our cells eventually just can't do it anymore. So we get all wrinkly and we just can't. Well, stem cells don't do that because they have an enzyme that keeps that length the same. So they can just keep on going forever. And our cells don't reactivate these enzymes, which is to say they are not immortal. They won't go forever. And yet they still appear to have OCT4, SOX2, NANOG. So we are really hoping that we are getting lucky, and this is nothing else but luck, of maybe finding a way to make a cell that is a potent cell but may not form tumors when put in vivo. And this is a point we have to address. There's very little difference between a cancer cell and a stem cell. And that's where cell transplantation encounters, embryonic stem cell transplantation encounters many problems. FDA, which is the agency that regulates all the therapeutics, in 10 years since they started using embryonic stem cells, allowed 2 clinical trials. 2. 11 years, this day and age. We should have hundreds. Because they cannot convince them that these cells are not going to start running all over your body making tumors. So you heal one thing, but you make somebody a liver tumor. That's unacceptable. You can't do that. And that is a huge problem in stem cell biology, how to control that they will never be stem cells again after you transplant them. Thanks for that question. I should have talked about that.
Še kakšno vprašanje? Zgleda, da smo publiko
They're saving themselves from next year. I just can't stop talking about next year. Thank you very much.
Thank you very much.
Thank you. It was a pleasure. Thanks. Pleasure. Thank you. Thanks for having me.
No, zdaj ste pa vsi skupaj vabljeni še na kozarček vina in klepet. Našo gostjo imate na razpolago, izkoriste priliko.
Obvezno.
Mi pa vsi skupaj upamo, glede na področje, ki je tako cvetoče in na tvojo zagnanost, sem prepričan, da se bomo v kratkem spet videli in da nam boš povedala, kaj je novega in katere korake ste uspeli narediti.
Pa bomo mogoče začeli korakati pa od tukaj, pa pol na oni strani, pa bomo skupaj prkorakali.
Mi bi si tega zelo želeli.
Se bomo potrudili.
Ja.
Sigurno. Hvala lepa.